be by the lactose fermentation or not. So here's our lactose showing again, or McConkey showing positive for the lactose. Bile salts crystal violet inhibit the positives. Again, differential is lactose. So things like E. coli, for instance, are lactose positive. On this one, we're showing down in this corner right here what a lactose negative would look like. So something like salmonella, for instance, and so it's not going to ferment lactose. You don't get the red color. Whereas the other three here are all showing a positive response for lactose fermentation. Now, the PDA is a selective for the positives and against the negatives. So phenylethyl alcohol is our selective agent. So anything that is gram-positive should grow. We did see, unfortunately, that some negatives do grow on there as well. But it is supposed to select against the negatives. Catalase, we have the, again, the H2O2 is our substrate, and when it bubbles, it's positive. It's producing molecular oxygen, also some water too. And so remember, everything is catalase positive except the streps. They are all catalase negative. Again, hydrogen peroxide is our substrate. The Staphytec thing we did the other day, again, it's an antigen antibody response. So these wells right here on the card have Staph aureus antibodies in them. And then when you mix the compatible antigen with the antibody, you get a clumping. And so this is designed to distinguish Staph aureus from any other staphs or anything else whatsoever. It doesn't matter. But you can see the particular kind of look right here. So that's our positive response right here. And that means that the one at number 3 with the clumping. So whenever you've got the antibody and its compatible antigen, they will bind together to produce a clumping effect. Whereas over here on this one where we had Staph epidermidis, it doesn't have the antigen isn't compatible with the antibody, so nothing happens. And it is the same as the blood typing sort of response. So the bacteria bring the antigens, because that's on the surface. The card has the antibodies, right? Right, oxidase, basically we're just trying to show if they've got cytochrome oxidase. So the oxidase reagent turns black color, like with Pseudomonas was positive, alkalogenase is positive for the oxidase response. And so again, just put the reagent on the bacteria on the swab. Blood augurs differential, so everything grows. We look for the effect on the blood, which is called hemolysis. The enzymes are called hemolysins. So on this one in particular right here, you can see that distinct clearing. That is a beta hemolysis effect. We have right here what we would call the gamma effect, because there is no effect on the blood itself. It's still growing there. Down here, we're getting a little bit of a partial beta effect. Okay, and then this is a gamma. I'm looking up, holding it up at the light. So again, you don't really see anything around the periphery right here of the colony. So it's growing, but no effect. So that's the gamma response. On this one, this is to show over here the alpha response, which gives us that kind of greenish yellow look. Strep pneumoniae is the one to think about for the alpha response, because it is the most obvious in terms of that response. The other two on the plate right here are both gamma, because again they're growing, but there's no effect on the blood. And this is showing the antibiotic response. So on this one, both of them are showing sensitivity. The thing about it, though, is when we do the bacitration response, it's a very low concentration as opposed to a higher one right here. You tell if it's sensitive by having, again, a clearing here around the disc. And strep pyogenes is sensitive to the bacitration at the normal amounts, whereas the strep pneumoniae, which is this one here with the alpha response, is usually going to be resistant to the low levels of bacitration. But this is a higher dosage right here, so we're seeing the response. So anyway, again, I want you to remember pyogenes, sensitive to bacitration. The pneumoniae is sensitive. This is the salt. So again, if it's transparent after growing, that means it's negative for salt. tolerance of 0.5%, where if it's cloudy, it is growing, so that means it's growing well, turbid look. So it is selected for salt-tolerant individuals. Mannitol salt, same thing, selected with differential. So we select for the salt tolerance. All 4 of these grow on salt. And then we look at how they affect the mannitol. Mannitol is the differential agent. So if it uses mannitol, we get a yellowish color in the agar. You get acidic there. It's phenol red again, turns yellow with acid. Whereas if it doesn't use the mannitol, then it just remains the same color in the agar. And again, it's still salt tolerant. So both of these salt tolerant and use mannitol. These 2 salt tolerant but do not use mannitol. Then the deal about the antibiotics and disinfectants. So you need to be able to look at plates and compare and see which one is the more effective or less effective. And so you will have the labels on there. If I'm asking about anything specifically, you won't have to guess. Everybody uses different stuff. But as you look at it, you know, you can see that the amount of clearing on this one, of course, is less than these others, so that's the least effective. The one that is the most effective looks to be this one down in here, because the clearing is wider than these 2 right here, kind of slightly less effective. And we just measure the diameter of the clearing to determine which one is which, you know, in terms of the more or less. But again, you'll have the labels on there. Now remember, we're comparing negative and positive for this, so you know, you may have something like this to look at, and I could ask you which one of these is more effective on the gram-negative or the gram-positive or whatever. So this is the one from the previous one. And so over here we've got the same stuff, except now we're on the E. coli. And so you can see that just in general, the antibiotics tend to be more effective on the gram-positive, at least one of these here on the gram-positive, than they are on the gram-negative. And you can see with E. coli that basically 3 of those basically had no effect on the E. coli, whereas over here they had some amount of effect on everything, and of course quite a bit of an effect with some of these. So you'll be looking at those side to side. And then here's the thing about the disinfectant. So again, you know, it's just a comparison to see which one is more effective on the gram-negative versus the gram-positive. You can see in our example right here that we're actually showing that the Lysol is not very effective whatsoever on the E. coli, either is the bleach, whereas we do have pretty good response to the with the Staph aureus with Lysol, but not so much there on the bleach. So anyway, it's just directly comparing the 2 forms. Bile-esculent, selected differential, so again, select for those that grow on bile. And we differentiate by the breakdown or the use of the esculent sugar. Again, black color is our positive response. We only have 2 things that are positive for bile-esculent, Strep bovis and Enterococcus faecalis. Those are the only 2 that are positive for bile-esculent. And here's the thing we did the other day. So again, with the UV, you know, if you've got shielding around the periphery, everything's fine. If you've got direct exposure to the UV, then you kill the bacteria. We do see some regrowth in the irradiated zone, which means either you've got mutation repair. They do have this light-dark mutation repair. We're not attempting to try to show it, so sometimes that happens. Sometimes you got so many bacteria there on top of your pile of bacteria that the ones on the top are killed, the ones below are not. So anyway, again, you see that. We also see it with the antibiotics and regrowth. Here's the one with the lid. So again, just showing that if you have the plastic on, it does shield no matter how many minutes you expose it. Here's the optogen now. So what we have again is the strep pneumoniae on this side, the pyogenes over here. So again, this P disc is the optogen disc, showing that this one is sensitive to optogen, whereas this one kind of grows up and out one side and the other of it, so it's not sensitive whatsoever. All right, and then for the hectone enteric, so again, it's selected differential, select against the positives, grow the negatives. We do have a orange to yellow color here for using the differential agent, which is lactose, but also has sucrose and salicylic, 3 things. Dark color like this means that it is not fermenting, but it's growing, so that is a negative. Down here we've got streak lines, so nothing grew, so the positive's not on there. We do see black coloration if it produces H2S. So things like Proteus, things like Salmonella, or Citrobacter for that matter, will produce a black color on the hectone, the pterygography. And then for the EMB, we have, again, selected differential growing. The negatives, not the positive. Lactose, again, our differential agent. So pinkish to red color is positive for lactose fermentation. E. coli, again, has this greenish tinge to it. When you look at it, it's fermenting lactose, but green right after, a day or two after it's inoculated, but it will turn pink. That doesn't mean it's fermenting lactose, and it's specific for E. coli. And anyway, you see right there, EMB. So, okay, that's our thing left for this unit. I don't think that we